Hello everyone, myself Priyanka Vijay Kulkarni. In this video we'll see about the blotting techniques. So basically in this video we'll see what is meant by the blotting and what are the advantages or the aim of this blotting technique. So basically this blotting technique, it is a very important, which is mostly used by the in the molecular biology and the biochemistry practicals. Yes, so basically this blotting techniques, there are the different types of the blotting techniques are there, but we'll see first what is mean by this blotting technique. So this blotting technique is basically, it is utilized to detect the different types of the biomolecules like the DNA, mRNA and the proteins during the gene expression. Okay, means what? During the gene expressions means conversion, or during the conversion of DNA to DNA, DNA to mRNA, mRNA to protein. What are these stages? How these biomolecules are going through these cycles? If we have to see or we have to detect, then we can use this blotting technique. Then another aim of this blotting technique is, as we learn in this topic under the advantages in the rDNA technology, means recombinant DNA technology. So it is helpful in the recombinant DNA technology. How? That the main aim of this blotting technique is to find out the position of cloned gene. Cloned gene means what? Cloned gene means the gene of our interest, which we cloned into the particular vector. For example, we'll see. Yes, suppose this is a one vector. Okay, and recombination means what? Combination of the two different types of the things. Here we'll take the two different types of the DNA. This is your vector. Okay, we can take it as any plasmid or any bacteriophage vector, and this is your gene of interest. Okay, suppose this is a insulin gene. Okay, and you have to insert this gene into this vector. So basically what steps you have to done? You have to first cut this vector, and this portion you have to ligate over here. So for this cut, there is a different types of the restriction sites are present on this vector, and this vector basically consists of the different types of the genes on that. So like different types of the gene, like the ORI, origin of replication, then antibiotic resistant gene. Suppose here we'll take the kanamycin resistant. Kanamycin is one type of the antibiotic, and these antibiotic resistant genes are probably present on this vector. Okay, so what happened that in this case, or in this vector, suppose there is a one antibiotic resistant gene like the kanamycin resistant gene. Okay, and in this gene, in between or within this gene, there is a one restriction site is present. Suppose this is a site of eco R1. Okay, this is a site of eco R1. So by using the eco R1 restriction enzymes are nothing but work as a scissor. Okay, as a cutter. Fine. So this enzyme will cut this vector at this point. So what happen? It will look like this. Okay, and this is your gene of interest, insulin gene. Okay, so this gene will come and ligate over here. Okay, and it will become a recombinant vector. Why recombinant? Because here there is a no gene of interest or no any insertion is occurs or no any disruption of this kanamycin resistant gene. But in this case, this EcoR1 enzyme, restriction enzyme, it identify the EcoR1 restriction site in between this kanamycin resistant gene, and it will cut over here, and this part means insulin gene will come and ligate over here, so it will become a recombinant vector. Yes, so in this recombinant vector, this is your gene of interest. So you have to find out the position of this gene of interest into this vector. Okay, so to locate the position of this gene into this vector, we have to perform the blotting techniques. Okay, yes. So what is the first aim? The very first aim is that you can detect the different types of the biomolecules, like the DNA, then the mRNA, and the protein. Okay, and the protein during the gene expression method. Okay, during the gene expression you can detect these types of the biomolecule. This is the first aim of this blotting technique. And second aim is to find the position of cloned gene. Cloned means your gene of interest. Okay, cloned gene in the recombinant vector. Cloned gene means what? Cloned gene means your gene of interest. These are the basically two main aims of this blotting technique. Blotting means a transfer. Okay, transfer of particular gene from one to the another. Okay, so yes, we'll see. It is nothing but the visualization of specific DNA, RNA, or proteins among the many thousands of contaminating molecules. Means from different types of the molecules we have to identify the particular molecule. Okay, so it is termed as the blot transfer. Okay, yes. Then there are the different types of the blotting technique. Okay, basically in your syllabus there is only 3 types are mentioned, but there are 4 or more types of the blotting techniques are present. First one is the southern blotting. Southern blotting means what? Southern blotting, the name, this name is come after the scientist Edward Southern in 1948 or in 1938. This scientist discover this southern blotting technique or detection of DNA technique. So the name after that scientist Edward Southern. So this is known as the southern blotting, means to detect the DNA molecule. Second is the northern blotting. Northern blotting, it is used to detect the RNA molecule. There is no scientist like a northern. Okay, there is only one scientist, southern, and after the name of that scientist the southern name is given, but northern there is not such a scientist. So northern is just a synonym to the southern. Western is also just a synonym to the southern and northern. Okay, so northern blotting basically it is used to detect the RNA molecule, and western blotting it is used to detect the protein molecule. Okay, there are the basically three types of the blotting techniques are there: southern blotting, northern blotting, and the western blotting. But there are some another blotting techniques are also there. For example, there is a one blotting technique like the eastern blotting technique. Okay, so in the eastern blotting technique basically we can detect the post translational modification. Okay, what we can detect over here? The post translational modification, like the glycosylation, addition of the lipid molecule to the protein. Translation means what? Protein molecule is produced after the translation. So post translational modification means what? to that protein the different groups are added, like the fat is added, then the some what we can say sugar is added. Okay, so that type of the addition or deletion we can detect by using the Eastern blotting. Okay, this is the 4 type Eastern blotting, which is used to detect the post translational modification. Okay, and the 5th type, there is another type is southwestern blotting. Okay, southwestern blotting. So southwestern blotting means what? Yes, it is used basically to detect the interaction of DNA and protein. Okay, it is used to detect the interaction of the DNA and the protein. It is known as the southwestern blotting. Okay, these are the different types of the blotting techniques. Many more there, but these 5 techniques are important for you. Okay, now we'll see what actually this blotting technique and what steps are involved. Okay, basically the first step in the blotting technique is, we can see here, the first step is digestion of your DNA or RNA molecule, whatever your sample is there. Okay, so digestion, it is a first step, digestion, or we can call it as a restriction. Okay, then second step is after digestion what happened. For example, I'll tell you with the example. Okay, suppose this is a long chain of the DNA. Okay, and first of all what you have to do, we have to cut that DNA into the small fragments. Okay, for this cutting we require the restriction enzyme, means Caesar. Okay, or the digestion material. So here the restriction enzyme is important. First step is digestion of DNA with the help of different digestive enzymes or the restriction enzymes. Second step, after digestion what happened, the different fragments of the DNA is prepared or it is made. Okay, so it is with the different length, so you have to separate that fragment. Okay, so for the separation of the fragment you have to perform the agarose gel electrophoresis. Okay, what you have to perform, agarose gel electrophoresis or polyacrylamide gel electrophoresis or SDS electrophoresis. Okay, by using all this electrophoresis technique you can separate the fragment of this DNA on this gel according to their molecular weight. Okay, after separation what is the third stage? After separation you have to transfer this, or you have to make a replica, okay, of this gel onto the blotting paper. Okay, third step is blotting. Okay, so there is a blotting paper is required. Suppose this is a gel, you have to put your blotting paper on this gel. You have to give some pressure in the presence of buffer. So what happened? This gel will do its replica means its copy onto that nitrocellulose paper or the blotting paper. Okay, and so this is a gel, you can copy, or you press this nitrocellulose or blotting paper on this so that all these bands are blot onto the blotting paper with the same position and everything is same. Okay, so this is your, yes, with the same structure and all these things. So this is your blotting paper. After blotting you have to do the probing. Okay, fourth is the addition of the probe. Why to add the probe? Because what you have to do, you have to find out the position of the clone gene. Okay, so to find out this you have to add a probe. Probe means what? Probe is nothing but the simple DNA or RNA chain, nucleotide chain with the length of 10 to 10,000 base pair long, or 10 to 10,000 bases long. It is nothing but the probe means what we can see. Probe means what? Yes, so probe means it is either DNA or RNA sequence, or DNA or RNA chain with the length of particular 10 to 10,000 base pairs. Okay, so this DNA, it must be complementary to our gene of interest. Suppose your gene of interest is insulin, so the proper nucleotide sequence of this insulin you have to know, because it is your gene of interest. So you already aware about this sequence. Okay, and you have to find the position. So what you have to do, you have to make a complementary sequence of this insulin. Okay, suppose this is insulin sequence ATGC. Okay, this is a insulin, and you have to find out the position of insulin in this vector. So what you have to do, how to make the probe? Okay, you have to prepare a DNA stretch or DNA sequence which is complementary to the insulin. Okay, so yes, what is the complementary to the ATACG? So this is your probe. Understood? Yes. So in this way you can prepare a probe which is complementary to your gene of interest, and in this way what you can do, you can add the probe in your blotting paper. Okay, you can add a probe, or you can dip this paper into the solution of the probe. Then what happened, our gene of interest and probe will attach to each other or will hybridize. Okay, there is a particular word which is known as the hybridization. Hybridization means what? Attachment. Okay, like this. Suppose 1 2 3 4, and this is your probe 1 2 3 4. Okay, so it will get ligate, it will get hybridized. Okay, yes, so probe and your gene of interest will hybridize. Then next step, what we have to do, and yes, there is a one important type. Whatever your probe, it is either radioactively or fluorescently labeled. Okay, why? For the detection purpose. Okay, so this is your probe. Okay, this is your probe. Yes, so this is your probe. It is either labeled with the radioactive or the fluorescent label. Okay, yes, this is your probe, this is your insulin molecule, so it is a complementary, so it will get hybridized. Okay, and after hybridization, what you have to do, you have to see this nitrocellulose or whatever your blotting paper under the autoradiography. Okay, why autoradiography? Because you have attached radio label probe to your gene of interest. So by autoradiography, what you can see, now suppose these are the fragments of DNA. Okay, and this is the fragment of which contain your gene of interest. Okay, another fragments are of the remaining vector. Okay, they don't have this gene of interest, only this fragment has the gene of interest. Okay, so under the autoradiography, what we can see, it you can see this is a shining or fluorescence. Okay, you can see the fluorescence band, and you can position that band, okay, in that vector. So you can see this is your gene of interest. So understood what is the aim of blotting technique and what is the procedure which are used over here. Yes, we can see by using one figure. Yes, this is the figure. Here we can see here this is the normal your DNA. Okay, first step is what? Digestion or cleavage or restriction enzyme, by using restriction enzyme. So cleavage with one or more restriction enzyme. After cleavage, what happened? Fragments of the small fragments of the DNA is produced. Now you have to separate that fragments according to their molecular weight and length. So you go for the electrophoresis process. After electrophoresis, what you have to do, you have to make a replica of that gel into the blotting paper. Okay, so for to make the replica, this is important thing. Okay, how to make the replica, you have to transfer this band to the nitrocellulose paper. For that purpose, there you have to take a one like this chamber. In this chamber there is a glass support. See, you can see here, this is a glass support. On that glass support, nitrocellulose filter is placed, and after that agarose gel is placed, and then there is a different types of the tissues are misplaced, and there is a weight is given. So in this way, the whatever this DNA sequence, it will get transferred to the nitrocellulose membrane or your blotting paper. So here you can see this and this is same, but this is electrophoresis gel. Okay, this and this is same, but this is electrophoresis gel, and this is nitrocellulose filter with the DNA fragment which is positioned identical in the gel. Okay, after that you have to do the probing. For this, you have to add the labeled either radioactively or fluorescently labeled probe. After the probing, you have to do the autoradiography, which shows the particular band with your gene of interest. Okay, the same thing is given over here in the color format. Yes, here the DNA is applied, electrophoresis is done, migration is occurs. After migration, what we can see, yes, migration is occurs. After the migration, you have to transfer it to the nitrocellulose paper by this way, sponge or with the help of wet towels. You can transfer this to the your nitrocellulose membrane or nitrocellulose paper, and the transfer is occurs by a capillary action. Okay, yes, then you have to do the probing. You have to add the probe, and which is complementary to your gene of interest, so it will go and bind over there. And you have to do the autoradiography, and after autoradiography, the autoradiogram is generated. You have to see that autoradiogram, and you can see these bands means this is your gene of interest. Okay, yes, so this is all about the blotting technique. Okay, what is mean by the blotting, or what is its importance. Okay, but nowadays there is a one thing, that nowadays rather than this blotting technique, the real time PCR is probably used. It is also used for the same thing, means for the detection of biomolecules, and second, to position the cloned gene. Okay, real time PCR is used, but this is also used, this is also important technique. In next lecture, we'll see about the sudden blotting. Yes, thank you.